Hi, my name is Iris, and today I'm going to be presenting on the Graduate Research Enhancement Award funded project that I did, which was creating a complete phylogeny using UCEs or ultra conserved elements for the Tenebriana genus Onymacris. So there are a lot of darkling beetles in the Namib Desert. Many of these belong to the subfamily Pameliinae, which is common in desert systems and very successful there. And we see a lot of variation, especially in coloration, elytral structure, body size, etc., as you can see from this photo below, in the tribe Atasmini, which Dr. Mary Louise Penrith really helped define. And she paid particular attention to this relationship between Onymacris, Eustopolis, and Physodesmia. So Onymacris comprises 21 species. There are 13 in the southern Namib sand seas, and then 8 in the northern Kunini and Skeleton Coast sand seas and dune systems. And white is in quotations here because white refers to pure white as you see on the screen or kind of tan and striped. And these are flightless coastal dune inhabitants, so they're very tied to these dune systems. They cannot survive without them. And there are a lot of unique adaptations specifically within this genus, including behavioral, such as fog basking, or morphological such as wax blooms, coloration, etc. So Penrith in 1984 created a morphology or a phylogeny based off of morphology. And she had the white and black species not forming distinct clades, but rather the white species were paraphyletic with black species. And then in 2012, Lamb and Bond came along with the first molecular phylogeny, and they were trying to really investigate that Eustopolis, Physodesmia, and Onymacris relationships, as well as look into the monophyly of the white clade. And they did establish the white clade as monophyletic. However, they did find that it was Onymacris as a whole was not monophyletic because Physodesmia globosa is sister to the white species. So the white clade is monophyletic in regards to black Onymacris species, but not a black species overall because Physodesmia is a black beetle. So the goals of this study were to create a really rich subgenomic phylogeny, really robust, to do some species delimitation if needed, because we did have samples from populations that had been previously weren't able to be included in molecular studies. We also wanted to evaluate the generic status and also have a rich data set for evaluation of traits and these unique traits we see within Onymacris within a comparative methods framework. So this is just kind of the preliminary part of what the study will eventually end up being. So for phylogeny, we selected 1 to 2 specimens per locality sampled, which totaled 72 species. And we were able to include all these subspecies and all known distinct populations for each species within the genus. And then we extracted DNA using QIAGEN DNA kits, and then we sequenced that DNA using the MyBates UCE Coleopter kit. And we're actually able to run more samples by deciding to do an in-library prep instead of outsourcing that. So we were able to save some funds by doing the in-library and then buy more probes in order to run more samples. And then the phylogeny was created using Figtree, or was visualized using Figtree, and then rate smoothed using PhyTools. So for our methods, we did have from previous population genetic studies some distinct populations that could have potentially been new species. And so we selected a species concept, which is a cohesion species concept, which says that a species is the most inclusive group that has genetic cohesion, so gene flow, or ecological cohesion, aka they occupy the same areas, have the same phenotype, niche, etc. To evaluate our populations that we wanted to investigate, we used a variational autoencoder analysis, or VAE, which is just a population genetic structuring analysis. And then for actual delineation, we use Delineate, which allows for the process of speciation that's still kind of occurring, and it estimates a tempo of speciation. So this is our complete phylogeny for Onymacris. Again, we were able to include all of those subspecies and populations that were not able to be included in past studies. So that was really exciting. And we had a lot of really well-supported nodes here. The only ones that didn't have high support, so 95% or above, have this red dot on them. And we see our white clade and our black clade falling out as we would expect them to, with Physodesmia still falling out as sister to white Onymacris species, which was also supported by Switchenberg et al. 2023, I believe. And this is really exciting because this gives us a very solid foundation in order to do comparative analyses of the unique traits that I mentioned before, like fog basking, which we see falling out in two different areas with bicolor and unguicularis, wax blooms, which we see across the tree and in several of the outgroups. So clearly some losses and gains in that trait, as well as coloration, which we see with that white clade. So for our VAE, we expect to see clustering of where there are distinct populations and distinct clusters and overlap where there's some ambiguous relationships. The color cloud is the standard deviation, and then those black circles are the mean. So here we see some of those distinct clusters and some overlap, especially within those subspecies groups. And actually, Langivisarii is one that we are going to look at in our delineation analysis. And then here we have two populations of Marginopennis that we're going to look at in the delineation analysis. And we can see there's a lot of overlap there with O'Brienii. For our black clade, again, we see some distinct clustering and then some overlap. Here, though, we did see some overlap we weren't expecting. So with Boschimanus and Boschimanus subunguata, we can see they are very distinctly separate, no overlap whatsoever, very clear, no relation, no gene flow. But we do see Boschimanus overlapping with multistriata. So that was also a population that we investigated in our analysis. So for delineate, it gives us this annotated output. The blue lines are species that we did not question. We have no doubts based off of the morphology, previous analysis, et cetera. Yellow are the yellow branches are the ones that are under investigation by the analysis. And then the terminal gives us the population designation followed by the actual current species name. The population designation either is going to give a already occurring population, or it is going to provide us with a novel species marker. So for our bicolor results, we had an Angolan population that's very isolated within bicolor. And through this analysis, we see that it did group together with bicolor and indicating that speciation has not yet occurred, even though there is such a great divide. It probably they've not been evolving separately long enough. So there are two subspecies that were called into question by the analysis. Langivisarii, which you see on the screen here, has a slightly different coloration than Langicornellii. However, in this analysis, it did have the population designation of being Langicornellii. So it is not a distinct subspecies, but rather a color morph within that species complex. And then for the Boschimanus, it has different elytral patterns to subunguata and multistriata. And in our analysis, it did group, as we expected, with multistriata based off of what we saw in the VAE. So it is not a subspecies of Boschimanus, but rather a morph of multistriata. There were two populations of Marginopennis that we analyzed, one that's Angolan, and again, separated, very isolated geographically, that's sister to O'Brienii at the bottom of the screen, and another one that was a southernmost population that we see at the top of the screen. And both grouped with pre-existing populations. So that southernmost grouped with Marginopennis, and that Angolan population grouped with O'Brienii, despite it looking identical to other Marginopennis species, which indicates that there's definitely some conservation of the phenotype that has occurred here. So in summary, we have a reduction of Onymacris from 21 nominal species to 19. We need to synonymize Boschimanus and multistriata and Langiviseriae and Langicornellii. And we also definitely are planning on going in and looking at the morphology here. Penrith did state when she was looking at morphology that it was hard to find characters just because Semophilic taxa tend to have very conserved morphological characters related to their lifestyle. So we are going to go in with morphometrics and traditional scoring methods to look at some further characters, specifically within the genitalia and the genital tract, which has not been looked at in super great detail, especially in recent years with some more modern technology. But we are planning on re-examining these characters and then creating a new key for the genus to reflect the synonymization as well as kind of the variation between populations that we see in some of our specimens, and additionally to examine that relationship with Physodesmia and resolve the monophyly from Onymacris. So Penrith stated in 1975 that there was incredible interest in Onymacris, and now we have a robust phylogeny and rich subgenomic data set in order to use to test our hypotheses. And the relationships in highly studied taxa really shouldn't be taken for granted. We do need complete taxon sampling and a variety of methods in order to make the correct inferences. So that study really highlighted that as well. And with that, I would like to thank my support, Dr. Trip Lamb, as well as 2 collection trips to the Namib Desert, and also my personal funding through UC Davis. But a very special thank you to the Coleoptera Society Graduate Student Research Enhancement Award that funded the bulk of this work, and especially buying those probe sets, and has really made this possible. And with that, I'll take any questions.